Hello, my name is Greg Cosgrove. I'm Chief Medical Officer of the Pulmonary Fibrosis Foundation. I'd like to welcome you to the current session entitled What's in the Pipeline? Current Clinical Trials, Potential Therapies, and How the Process Works. And so I'm joined by my colleague Bill Bradford, who is founder and CMO of Therapeutics. Probably many of you don't know Bill Bradford, but you know of his work. He's been key and instrumental to the development of several therapies in different therapeutic spaces, but for this group, he was instrumental in the trial design and redesign of the therapy termed pirfenidone, or now what is Esbriet, and it's through his work with the team of individuals that they were able to better understand the difficulties of evaluating therapies in idiopathic pulmonary fibrosis, which was, I thought, alluded to this morning, given the complexity of the disease, the heterogeneity of response to some therapies, and the variable course, which in designing clinical trials is challenging to say the least. So as a testament to his expertise, and I think moving forward, thought it would be an excellent opportunity to educate those of you who may or may not understand the clinical trial process, because I think it is very helpful perhaps for you to understand that if you're participating in trials or considering to participate in trials, but also then perhaps lead into the numerous trials that are currently ongoing in the field, which I think is exciting because it suggests, number one, incredible interest to try to help patients. Number two, the diverse possibilities for multiple different therapies as we evaluate them, to perhaps helping individuals with pulmonary fibrosis, and perhaps some of the challenges as we move forward. And it was perhaps suggested in the prior talks this morning, so we'll move forward and discuss those in a little bit more detail. But just to set the expectations, it's highly unlikely that we can go through the 134 clinical trials ongoing right now based on clinicaltrials.gov. So broad strokes to give you an idea, and certainly you can ask questions after this. We'll have Dr. Bradford give us his presentation. We'll take time to take questions because I think that's an important aspect of about understanding clinical trials, and then afterwards we'll go through a little bit more of the pipeline and help to understand not just clinical trial design in general, but perhaps some of the challenges we have ongoing and specific to idiopathic pulmonary fibrosis and opportunities as I would look at it. So I welcome you, and I'd like to introduce again Dr. Bill Bradford.
Thank you, Greg, and welcome everyone. It's a real pleasure for me to be here and have the opportunity to talk to you guys about IPF clinical trials. 134, I remember probably the first few years I was working in IPF, which was 15 years ago. We could certainly count the number of clinical trials on one hand, so it's really been a phenomenal and exciting transition that we've had. So in addition to being an executive and the chief medical officer at Therapeutics, I'm involved with a number of companies that are doing work in the IPF space. As far as my disclosures, 2014, I'm sure you guys are very familiar with what happened then, but it was really a landmark year for IPF. The phase 3 trials, that's the last stage of clinical development of pirfenidone and nintedanib, were presented and published in the New England Journal of Medicine. These studies both represented just many, many years worth of work and had very conclusive outcomes with respect to compelling efficacy, that's effects that benefited patients in addition to the safety. FDA, in reviewing the applications, immediately recognized the gravity of the IPF disease state and the strength of the applications, the clinical data, and expedited their review of those applications. And in October, both these drugs were approved, and really a landmark for IPF, where we finally had 2 drugs approved, which is a great thing for patients, and I think it's also a great thing for the field, because if one looks across drug development in different therapeutic areas over the years, Once there's a regulatory approval, it defines a pathway with a regulatory precedent. And then all of a sudden, that disease state, IPF in this case, gets very attractive to a lot of other companies that are interested in developing drugs because there's no longer risk about what the pathway is. There's a clear path. And so that really happened in 2014. And the 134 is in part a response to having that defined regulatory pathway. And so today, I'm going to talk about that pathway, the pathway that was followed by pirfenidone and nintedanib, and that's being followed by these many other drugs that are currently in development. I'm going to start with a very basic cornerstone here of my talk, and that is what is a clinical trial, just so we're all aligned on that. It's a research study in which human subjects are assigned prospectively to an intervention. This is also often done in a randomized fashion, such that groups that are on different therapies have relatively equal characteristics so that they can be analyzed that way. That's a word I'll use a lot, randomized, as we go through some of these studies. And then double blind, another important concept in clinical studies, meaning that the research subject and the experimenter do not know what treatment assignment folks are on. Are they on placebo or are they on active drug? That's unknown. And then folks are followed through over time and outcomes are assessed, safety and efficacy being the common ones. Out of curiosity, could I ask whoever has been involved as a research subject in a clinical trial, could you raise your hand just so we have a sense? Okay, we have a number of veterans in the crowd here, and that's good to see. So the drug development process can be split up into a series of stages that starts preclinically, and I'll walk through in some detail and show you some examples of these different stages. Then it goes through what we call an IND, which is investigational new drug. This is basically an application to the FDA. FDA decides whether the drug is ready to move into clinical testing. Goes into the clinic. There's 3 phases of development there, phase 1, 2, and 3. In general, as one progresses through 1, 2, and 3, the studies get larger and longer, and the focus begins to shift more towards demonstrating benefit on outcome measures that are important to patients. The NDA, that stands for New Drug Application, at the end of the clinical development, that's the application that goes to FDA. There's similar applications that go to other regulatory authorities globally to get a drug approved. And then following a drug approval, the development's not over. Usually there's phase 4 studies that are done, and we'll talk a little bit about those today as well. Jim Kiley this morning in the keynote address, if you had a chance to hear him, made I thought a very nice point about the importance of the community ecosystem to research efforts in IPF. And I think it's very important to highlight that here as we talk about developing drugs and designing clinical trials. This is a team effort. It really, it's a massive undertaking, and it takes a lot of different parties. Obviously, patients are front and center in that. We develop drugs for patients. We couldn't develop drugs without patients. And I'll comment on a few of these here. Regulatory authorities, obviously, the FDA has a mandate to ensure the public health of America, right? So they implement that in part and through the clinical development process and regulating what trials happen and what drugs get approved as well. I think an understated part of what FDA does is they can provide tremendously valuable information and guidance to folks that are running trials, how to design studies, different things to think about. It's really a tremendous resource there. We have patient groups and families, obviously integral to the support of the patient community and often quite involved in these issues around drug development. Sponsors, that means that the folks that are running the trial, they pay the bills typically, they design things, they manage it. So this might be NIH, it might be a pharmaceutical company, it might be an academic institution. And then lastly, I'll just mention the site personnel. When we go out and do these large phase 2 and phase 3 studies, it's absolutely critical to have a strong team at every site. And They're often the unsung heroes in these studies, because that's where it really all happens. The patients come in, there's the coordinators, the study nurses, the docs, et cetera, and that's an integral part of getting these studies executed in a high-quality fashion. So let me start with the path to the clinic, just to give you a sense for where this all really begins. So preclinical research, we talk about discovery research, which is the first part. This is where drugs are discovered. And there's really 3 steps here. The first is understanding the disease, trying to have some sense for the pathways that result in the disease, in this case IPF, and then try to identify targets in that pathway that one can drug. Okay, so for example, here you can see something called TGF-beta. That might be a target that one goes after, and then you come up with drugs that attempt to hit that target and modify the activity of that target. And there's a very sophisticated technology that has evolved now using structure-guided, structure-based drug design. And you can see the image here where it's basically a 3-dimensional target, and you can see the drug binding with that. So we often forget about what happens before we get into clinic, but there's a lot of work, a lot of very sophisticated chemistry, biology work that happens that supports drugs moving forward. Once you have a potential drug, you move into preclinical research. And this is the stage where you optimize your drugs to do the things you want them to do. For example, if you have an oral drug, you want to make sure it's getting good concentrations in the blood, for example. So you can try different drugs to see if you're achieving that. Then you start to put them into models. In the case of IPF, we have a series of models that we often look at. Typically, mice are used quite a bit here to see if a drug looks to be active. Do a lot of things there to predict, is this drug likely to help patients with IPF? And then there's a tremendous amount of work that happens looking at toxicities. And a lot of this is mandated by FDA and other regulatory authorities to ensure that the drugs are well characterized that we put into humans. And if there are safety issues, then they obviously won't move into humans. So all this work takes place, a lot of it in test tubes, a lot of it in animals. And if a drug is successful in preclinical research, the decision is made, let's advance this drug into human subjects. And there's a number of gatekeepers as one goes to advance that drug, as there should be. Here in the United States, and incidentally, as I talk about the regulatory authorities here, I'm going to talk about the U.S. because that's where we are, but really every region has different regulatory authorities and often different rules as well, but I'll be very U.S. focused here in my comments. So when you're ready to advance, you put an IND, investigational new drug application in, and FDA reviews it. This IND summarizes basically everything you've ever done with the drug. It's a very deep, comprehensive document, how the drug works, what all the safety findings are, et cetera. One also put forward a very detailed clinical plan. Here's what the clinical trials are going to look like starting at phase 1, the first study that you do. All this goes in and FDA reviews it. They review it with an eye first and foremost to their mission is to protect public health. And so it's very focused on safety and what are you going to do in the clinic? Is that going to risk any patient's well-being, for example? And FDA can either allow the application or disallow it or modify it. And all 3 of those happen with some frequency. And just to give the patient perspective, FDA is not the only safeguard here. So there's 2 other processes that one needs to go through before giving an investigational drug to human subjects. One is Institutional Review Board, and this is basically an appropriately composed group that reviews these applications, reviews the protocols, and says, is this safe to put into humans? And really, their job is to protect the welfare and health of human subjects very specifically. And again, they can say, nope, this is not safe, we're not going to move forward with this. And then the other check in the process is something called informed consent. And without getting into a lot of details, this is basically it's a document that summarizes everything a patient needs to know to make a rational decision about whether or not to participate in a study. And this includes the expectations: how long am I going to be in the study? How many times are they going to draw blood from me? Those kinds of things, and obviously the risk that's associated with participation. So once you've gone through all that, then you head into clinical development. And so let's talk a little bit about phase 1 clinical studies. So these are typically done to look at safety and tolerability, very important early on here. Explore the dose and the concentrations of drug. How much drug is in the blood? How much drug is in the lung? Those kinds of things. So it's not are we affecting lung function at this point? This is all very early on. And does the drug hit its target that it's designed to hit? These are typically small studies, 20 to 100 patients. As we go through these stages of development, I'm going to share with you some industry benchmarks, which are some data that have been collected looking at drugs in all different therapeutic areas, all types of different diseases, just to give you a sense for some of the key metrics. So here for phase 1, for example, time, the average time from starting phase 1 to getting to phase 2, 1.7 years, cost is $25 million, and the likelihood that the drug will advance. So if a drug comes into phase 1, how likely is it to go into phase 2? So that number is 64%. So here, roughly 2 out of 3 drugs will go from phase 1 and successfully enter phase 2. A third will not move forward for various reasons. And so I'm going to briefly give you some examples of different studies as we move through here. And the first study is always an exciting time. We call it the first in human study or the first in man study. It's the first time you've taken a particular drug and given it to a human subject. It's usually healthy subjects. These are usually conducted in special units where folks are monitored very carefully for any kind of safety issues. You start with a very small number of patients. You start with a small dose. You move very carefully up in doses to make sure they're well tolerated. And again, you carefully monitor safety, labs, and again, the drug concentrations. You can see the schematic on the right side of the slide there, and this is what a lot of these first-in-man studies look like. These are dose escalation groups. So you basically start and you'll expose maybe a half dozen subjects to a drug, and if they're okay, then you go to another half dozen subjects, and you keep going up and up until you get to the dose levels that you want to interrogate there. And again, that's almost always done in healthy subjects. Here's an example of a phase 1B study that was actually done in IPF patients. This is from Galecto Biotech, and I think this is a very exciting and informative study. So this is an inhaled drug. They did a 14-day study in 24 IPF patients. They had 3 dose groups, and they did something called bronchoalveolar lavage. You basically put a scope down in the lungs and you collect fluid and cells and can look at them. And if you look at the top right panel there, you can see 3 bars. So along the x-axis, we have the different dose groups, and the height of the bars are showing the drug concentration. So this shows exactly what you want. So this is an inhaled drug. They're going down in the lung, and they're saying, as the dose goes up, we're seeing higher concentrations of the drug. Okay? That's exactly what you want to see. The bottom right panel there, you can see a number of points. This is something that we like to do a lot. It's called PK/PD, but it's a way of looking at the relationship between how much drug you have in the lung and what the biologic effects are in the lung. This type thing is very important in early studies. So this is an exciting compound here from Galecto that will be moving forward in development. Phase 2 clinical studies. Again, the objective, safety and tolerability, still very important. In phase 2, you start to explore dose and you start to explore efficacy. These are not definitive studies where you can say this drug works, okay? This is an exploration phase still in phase 2. Larger studies, and I'll show you some small and some large examples, 50 to 500 patients. Phase 2 is the gauntlet in drug development. It's where the most drugs fail, has the highest likelihood of failure. Again, these are bigger, longer studies, 2.5 years on average. Cost is $58.6 million for a phase 2 program. If that's seeming high, you haven't seen anything yet. Wait till we get to phase 3. And again, it's the toughest stage of drug development statistically. So 32% of the time, drugs will advance from phase 2 into phase 3. So 2 out of 3 drugs don't make it. To run these big studies, as you can imagine, if you're spending $50 or $100 million, you need a lot of infrastructure and a lot of operational efforts to make things work to ensure success. And I won't go into detail here, but there's a massive sponsor team that needs to be in place with a lot of different skill sets. The team at the site needs to be strong. They need to be trained. You have to work through contracts, IRB approvals. A lot of these studies have all kinds of committees, steering committees, safety committees, endpoint committees that all need to be put together and chartered. And then there's all kinds of details around getting drug to sites in a big global study. How do you randomize patients? Who's monitoring the data to make sure it's high quality, et cetera. So just to underscore, this is a massive, massive undertaking to successfully execute these types of studies. Here's an example of what we call a phase 2A study. So this is kind of an early phase 2 study. Inhaled cromolyn for cough in IPF. And I really like this study. This was just published recently because it's going after a symptom that is of immense importance to patients here, cough. It's randomized, double-blind, cromolyn versus placebo. And this is a crossover design, which is a pretty slick little tool as you go to design studies. And in essence, if you look at the figure at the bottom, there's 2 arms on the left there. So half the patients will start out on drug, and the other half will start on placebo. Then after a period of 2 weeks, they will switch. And there's some real statistical advantages to doing that with being able to compare data across groups, et cetera. And so this was a clever way of designing this study. There were 24 patients, and the primary endpoint, which is the main outcome measure that one looks at here, was average daytime coughs per hour. And when one looks at the results here on this figure, so the y-axis, the vertical axis, is the number of coughs per hour, and you can see at baseline that's around 50 coughs. So really a profound rate of coughing. And then you can see at 7 days and at 14 days, assessments were done, again, looking at the number of coughs per hour. And you can see that there's a nice drug effect here at day 7 and day 14, where patients on cromolyn had less cough than the patients on placebo. This is still early preliminary data. We always have to be careful not to get too excited about phase 2 data, because history has shown that it doesn't always repeat itself. Is always an integral part of these studies is looking at safety or adverse events, as you will, and in this particular case, a very nice safety profile associated with the inhaled cromolyn. After phase 2A, we go into 2B, and 2B in many ways are, to me, the most challenging studies in all of drug development. So you're going from these smaller studies into a very large study, and you're saying we need to figure out what the dose is. We need to figure out if this drug is going to affect outcome measures that we can use in the phase 3 study. Boehringer conducted the Tomorrow trial of nintedanib, and I think this is an incredibly robust study and a great example of what a phase 2B study should look like. It answered the questions that needed to be answered. Large study, 430 patients, randomized nintedanib versus placebo. They interrogated 4 different doses of nintedanib here, and we'll go over some of that data. Primary endpoint was annual rate of FVC change, and they looked at a number of other outcome measures as well. So here are the FVC results by nintedanib dose. And on the y-axis here, we have annual rate of FVC change, and across the x-axis, we have the different groups. And so the beauty of this study is you understand the relationship between the different doses and the outcome here. And so obviously you can look at the placebo group in red. They lost close to 200 mLs on FVC. And you go to the far side, the highest dose, 150 milligrams twice a day, and those patients lost quite a bit less. That's the most striking result here. So that was the most efficacious dose. Then one can go and look at the safety, the adverse events, in the same fashion. And you can look at placebo, your reference group, versus the different dose groups. And here in the case of, for example, diarrhea, which is a side effect well associated with nintedanib, as you go from left to right here, you can see the numbers increasing. That's what we call a dose response. These type findings are really helpful as one goes to choose doses and then go on to the next study. And IPF progression, for example, most of the way down the chart there, again, you can look at that across doses, and you see that when you get to that higher dose that had the best findings on FVC, in fact, there's less IPF progression in that group. So you can look at both safety and favorable outcomes when you're looking at this type of data. So a great study, great result. This teed up the phase 3 program for nintedanib that went on to be approved. So on to phase 3. Really, when we think about objectives here, the primary objective of phase 3 in many ways is to support drug registration. And to that end, before one initiates phase 3, there's usually interactions with the various global regulatory authorities to make sure that everybody's aligned. If we do this study and we win on the endpoint, we'll get approved. And efficacy is a very big piece of that. It's also larger patient numbers, so one continues to focus on safety and tolerability as more and more data comes in. Back to our industry benchmark. So the time, 2.6 years, 2.5 years to conduct these. The cost, $255 million here. And again, this is not IPF specifically, this is across the board, but I can tell you today, $255 million is probably not far off of what it costs to run a phase 3 program for an IPF drug. 60% of the time, the studies advance to the approval process. An example of a phase 3 study is the ASCEND trial of pirfenidone. This was randomized, double-blind, pirfenidone versus placebo. Dose-ranging work had already been done, so there's only 2 groups in this study, the pirfenidone at 1 dose versus placebo. It was in patients with a confident diagnosis. You know, it's worth pointing out here that the issue of enrollment, which is one of the great challenges I think we face in IPF drug development. So this study enrolled 555 patients. To get to that, 1,562 patients were screened. So for every patient that was enrolled, 3 had to be screened. If you look at other industry benchmarks, to get to that 3, you need to start with 10. And it just highlights how hard these studies can be to enroll. So you start with 10 patients to get down to 3 that you actually take through formal screening, to get down to 1 that you actually put into the trial. So these studies can be very challenging to enroll. Primary endpoint here, FVC change. You're probably familiar with some of this pirfenidone data. Here on the y-axis, this is the proportion of patients that have a 10% decrease in FVC. That's a metric that has been shown to be very clinically meaningful. And if one looks at it week 52, you see that pirfenidone basically reduced by 50% the proportion of patients that had that bad outcome. And likewise, large phase 3, lots of patients, great time to look at safety outcomes here. And one can look, for example, at nausea, a side effect well known to be associated with pirfenidone. So there's more nausea in the pirfenidone group than the placebo group. Likewise, you go down towards the bottom there and look at IPF, and there's less IPS progression in the pirfenidone group than the placebo group. So this safety data, when you get to these large studies, is really telling, particularly for the more common adverse events. And then briefly on mortality. IPF is a fatal disease, and the regulatory authorities and really everybody's in total agreement that it's important to look at mortality as an outcome measure here. And here are the results from the ASCEND trial. You can see roughly 4% versus 7.2% of patients died of any cause. And because these numbers are kind of small, we often pull them with other studies. And so on the bottom there, you can see the results that were pulled with the capacity trial. And this is a very important outcome measure when one thinks about drug approval and frankly, just the relevance of a therapy benefiting patients. After one goes through phase 3, if they have a success, they go through the approval process. And the sponsor submits an application that includes an unbelievable amount of information, thousands and thousands and thousands of pages. And it's all very clearly spelled out what needs to go in that application. FDA reviews it. They can take up to a year. I think in general in IPF, FDA has been very friendly. They recognize the unmet need here, and they've expedited these applications. And then at the time of approval, they issue the prescribing information, which is a nice summary of the outcomes of really everything about the drug that FDA thinks the prescribing physicians need to be familiar with. So we've gone through the approval process, and let's look back at these industry benchmarks just to kind of add it all up. So on the time side, the synthesis to phase 1, 2.6 years, phase 1 to approval, 8.1. So overall, on the benchmark side, 10.7 years from start to approval. So incredibly long process. With respect to cost, as I think you well know, not all drugs work. And so the question is, what does it cost to get an approved drug? So to get an approved drug, it's not just the cost of developing that one drug, it's developing all the drugs you have to get one winner. So you can say maybe we got to start with 8 or 10 so that we have one that gets across the finish line, and that's how the health economists typically look at cost of drug development here. And you can look at the blue bars on the slide here, and this is the out-of-pocket cost. This is how much the sponsor pays to run these trials. So you can see the numbers for the preclinical, clinical, and total there, with the total coming in, I don't have my glasses on, over $1 billion. And if you've had a mortgage or a loan on your car, you know that money's not free. You have to pay interest. And so if you look at the capitalized cost of these numbers over this period of time, obviously the numbers go up substantially more based on that. Suffice it to say, it's a long and expensive process to get a drug through development. Following approval, there's typically additional studies that are done. These studies can take all types of different forms with different objectives. It could be safety, it's to characterize the way the product's being used, potentially new uses. Here's an example of the Passport Registry that was conducted with pirfenidone in Europe. So basically the European authorities, when they approved pirfenidone, asked for a registry of 1,000 patients, real-world experience. Take 1,000 patients, put them on pirfenidone, and watch them over time. Very simple study design. Gives a lot of safety data, real-world safety data. You can look at how well patients tolerated the drug, how many dropped off, those kinds of things. Here's some new data out of Beringer that was recently presented and published. So a combination study. We've come a long way now. It's fantastic to think that we're actually doing combination studies in IPF. It wasn't long ago where we didn't even have a drug to use, right? And so this was a study focused on safety, drug concentrations, and FVC. And basically, they took patients that were on a stable dose of nintedanib, and they randomized them to add pirfenidone on or nothing else on. So basically, at the end, you had nintedanib versus the combination of pirfenidone and nintedanib. And they were on this for only 12 weeks, so not that long, but it gave an opportunity to look at the adverse event profile. And we know both these drugs have gastrointestinal side effects, and that was in fact observed here, and it was worse with the combination. And then some enchanting data that is early and needs to be confirmed, so we shouldn't get too excited about it, but it certainly appears based on this data that there's less loss of lung function on the combination than there is in the nintedanib group alone. We know very well that nintedanib alone is much better than placebo. So it's an enchanting thought for the future that needs to be substantiated. But very encouraging, I think, particularly as we have a number of drugs moving forward in development. Opportunities for innovation. A few last comments here before I turn things over to Greg. So the number of drugs in development in IPF is soaring. Greg gave you some numbers. It's really impressive. We've had great news this year from a number of different companies on a number of different drugs that have been looked at here. All very exciting. But the question comes up, how do we accelerate this process? I just went over some of these timelines. It just takes so long to develop these drugs, and the more drugs we have in the pipeline, the more competition there is. There are things we can do, and I want you all to know that there's a lot of smart people working on these things, and I think patients also have an important role in this process. How do we measure FVC? This is a great one. So it's the established endpoint. FDA has used it to approve drugs. It turns out that you can measure it with handheld spirometers at home. And so the question is, does that give you any advantages? So the data to date suggests, it's not established yet, but suggests that we can use handheld spirometers that are very convenient for patients. You don't have to leave your house, right? You can do this at home. And you can get daily measurements or multiple measurements a week compared to when you're in a clinical trial, you usually get one every 3 months. And so it's a much richer data set. And it suggests that we might be able to do this in clinical studies, and we'll need less patients per study to get a good understanding of FVC. This is still, there's work to be done, but very encouraging so far. Mobile technologies, obviously they've infiltrated every aspect of our lives, and they're doing that in drug development, clinical trials as well, and really getting widely adopted in a lot of ways in other different therapeutic areas. On the enrollment side, we can create networks where patients can be very aware of trial opportunities. We can, for consenting patients, we can use data in these apps to identify candidates for certain studies. And as I mentioned with the home spirometry, there's opportunities for remote data collection. We can also use these for patient-reported outcomes, for example. All is very intriguing, and we need to move in this direction in IPF. And lastly, there's a lot of opportunities for patients and patient groups in the clinical trial process, including making the process more efficient. And I won't go through this in any detail. Let us say on the front end, we need to involve patients more. Patients have the insights into the disease burden, what matters to patients as far as outcome measures. We need to involve them more in the patient groups and designing clinical studies and help us work through these issues around enrollment and then interpreting the results of these clinical studies. So I think it's been widely appreciated by FDA and in this case, the Clinical Trials Transformation Initiative, a group that was formed to really increase the quality and efficiency of clinical studies. It's widely recognized that we need to move in this direction and be much more patient-focused. So with that, I will end. And Greg, do we have time for a few questions? Or? So I can take a few questions if, yes.
I find it very curious, both of these drugs were approved at the same time. Can you explain how that happened?
I think it was a combination of when Intermune and Boehringer were doing the studies and when the applications were submitted, and it's very clear that these were both in FDA's camp for quite a while, and they obviously made an active decision to approve these on the same day. Which is fine. Hey, we've been waiting for 20 years for one, and then we got two.
You said there was a bunch in the pipeline, drugs in the pipeline now, and if it takes 10 years from start to finish, how many of them are 7+, 8 years in the making? I mean, so are we expecting a bunch soon, or we're still 8 to 10 years away?
Well, it's 10 years from synthesizing a new compound in the lab to an approval, and I think Greg will probably go through this some, but I think there's some very good opportunities that are currently in the clinic today. Some of the phase 2 data that's come out even this year. What I think has happened, and this happens a lot when one looks across different disease states and drug development efforts. There's usually the first generation drugs that come in, and they usually work reasonably well. Once they have a pathway, then a lot more companies come in and they get a lot smarter about the targets to go after in that disease and understand the disease better. So typically the next generation drugs are better, and they're more likely to move later in development. So I'm actually very encouraged about where we are today and the number of drugs that are in the pipeline, including phase 2. It still will take a while, right? I mean, phase 3 is going to be 2.5, 3 years, and then another 6, 12 months before it gets on the market. So we're talking years, but it's not 8 to 10, I don't think. It's hard to predict, but there's some great drugs that are in the clinic today.
This process you just described, that's for drug development. How does that differ with the genetic engineering that's in the process right now, where they're growing your own lungs from your own genes? How does that process for approval differ from the drugs?
It's quite different. And I'm not an expert in that, but in general, the FDA has a drugs group and then more of a biologic type group that will deal with these cell therapies and gene therapies. And so it's a very different pathway. And obviously the science and biology is all very different as well.
In some diseases, the diseases get smart and they outsmart the things that are supposed to kill them. Have you found any of that in the trials that you've done, that IPF has gotten smarter and outsmarted the...
Yeah, you're exactly right. That's been a problem in a lot of disease states, oncology being a classic one. We have not, and you could ask the question why, and there's probably a variety of answers to that. But for what it's worth, the 2 drugs that are approved, they're not a molecularly targeted therapy that hits one thing. Both nintedanib and pirfenidone do a number of different things, and it might be that they're kind of protecting themselves from that problem because they do a number of different things. It might also be just that the biology of IPF is such that it doesn't adapt to kind of get around those particular drugs. We don't know the answer to that yet, but so far it doesn't seem to be a problem.
On the couple of drugs that you're excited about that could be a few years away but not 10 years, what exactly do those drugs do towards IPF?
Yeah, I think I'm going to defer that to Greg because it's probably time to move on to his talk, and that's the topic of his talk. I will say we're a lot smarter. We've learned, Intermune, we licensed pirfenidone in 2002. I'm not sure what year they started with nintedanib, but this was well over a dozen years ago, all this stuff. And we've had, as you can see from this conference, there's 1,000 people here. I mean, 10 years ago, there would have been a handful. So many smart people today are working on this disease state that we've learned a lot. And a product of that is we understand the biology better, and we can go after targets that make a lot more sense and are much more likely, in my mind, to work. And so I think we're moving into a whole new era here. Yeah, I'm sorry. Pirfenidone was licensed from another company, then it wasn't licensed for approval. Pirfenidone was licensed, I think, in 2008 or 2009 in Japan for use, and Intermune got approval, was it 2011 maybe? It was approved in Canada and in Europe before the last study that I showed you there, the ASCEND study that led to the approval in the US. And that's a whole nother long story. Thank you.